Να ανακοινώσω στα ελληνικά, γιατί είναι στα ελληνικά από ό,τι καταλαβαίνω και η ομιλία της. Είναι η κυρία Φουντή, η οποία θα μας μιλήσει για τη γενετική επιδημιολογία, νέα εποχή στο γλαύκωμα.
Καλησπέρα σας. You will allow me to say that in Greek, please. Actually, my lecture is in English, because the president of the meeting has asked me to do that. But I'd like to say in Greek that είναι εξαιρετική τιμή για εμένα να βρίσκομαι εδώ. Σας ευχαριστώ πάρα πολύ. Θα ήθελα να ευχαριστήσω και την Ευρωπαϊκή Εταιρεία Οφθαλμολογίας για αυτή τη δυνατότητα και την Οφθαλμολογική Εταιρεία Βορείου Ελλάδας που με πρότεινε για την παρουσίαση και την Ομοσπονδία Οφθαλμολογικών Εταιριών που με εξέλεξε. Σας ευχαριστώ πάρα πολύ. So, the topic I chose to present to you today is that of genetic epidemiology and whether it represents a new era in glaucoma. And in this context, I'm going to talk to you about possible changes in medicine in the future, about the genomic promise for glaucoma and what it means, whether there is any evidence to support that such a concept is actually realistic, and what is the current status of genetic epidemiology in glaucoma. So, a few years ago, faculty members of the Johns Hopkins University were asked to answer the question, how will medicine be practiced in 10 years? And to be able to answer this question, they thought about the current events and trends which are likely to affect medicine in the future years. And these are genomic research, the aging of the population, healthcare economics, the importance of public health, but also advances in technology. And based on these trends, they came up with a new curriculum for medical school, which is called Genes to Society. And this is the curriculum that medical students now follow at Johns Hopkins. So according to this curriculum, the context of illness and disease need to be reframed, and patients should not be seen as people who either have or do not have a disease, but they should be seen along a continuum from asymptomatic to terminal illness. Also, each patient's phenotype should be determined by a number of internal factors building up from the genes to the molecules, to the cells, the tissues, organs, and individuals, and of external factors, which is environment, family, and society. And ultimately, the goal would be to prepare medical students for an era when patients will expect individualized medicine and physicians will have the tools in hand to provide such care. But what does individualized or personalized really mean? Because medicine has always been personalized. We are treating persons. We're not treating crowds of people. Until recently, for the clinician, the genetic data were usually limited to family history or race and ethnicity, as this is self-reported by the patients. And also the terms genetics and genomics were these difficult terms, usually related to basic research done by someone else to determine the pathophysiology of the disease. But with this context, with this concept, the idea is that genomic information would be available for the physician to use for decision-making, and this decision-making refers to preventive treatments, but also interventional treatments on the basis of individual characteristics. And that would be perfect for a disease such as glaucoma, which is complex. It means that it comes from a combination of genetic and environmental factors, because these diseases have risk factors which can be different from one person to the other. The disease occurrence may be different, the disease progression may be different, and response to treatment can be different. So according to this approach, we should be able in the future to effectively screen people and tell them whether they are at risk for the disease, to establish a precise diagnosis at a molecular level, to predict rate of progression, and to predict response to treatment. But none of this is a reality yet. We don't have any of these tools yet. So how do we know that this approach is even realistic? It might be realistic because we already have a lot of information about the genome, because we have a lot of advances in bioinformatics and in statistics. And because of these tools, there has been a lot of research with candidate gene association studies, which is a very useful but a bit of an older approach. And these large genome-wide association studies where hundreds and thousands of genomic variations called SNPs are tested for associations with disease or traits in hundreds or thousands of people. And the difference between the two is that these first studies are a bit biased because they are looking at predetermined genes, where the other studies are unbiased because they look at the whole genome. And based on that, we do have personalized medicine for complex diseases, only not in ophthalmology. We are able to identify individuals at risks. For example, individuals at risk for breast cancer or at risk for cardiovascular events based on genotypes which seem to modulate cholesterol. Oncology is the typical, maybe the first and most typical example of a field of medicine where diagnoses are made at a molecular level and they can decide the prognosis of a disease. Pharmacogenomics, which is the study of how genes can affect response to treatment, and it's one of the most direct applications of genomics in clinical practice. In fact, more than 2,000 genes are now listed in this big database, which is available online. Anyone can access it, and you can see here, that was just yesterday, there is these new guidelines for treatment of cystic fibrosis based on this genotype. And you can also see the list of well-known pharmacogenomics associations and clinically relevant summaries, which means that this is not basic science. This is clinically relevant. This is for the clinicians at other fields of medicine. In oncology, it is possible to do genetic analysis for some tumors and predict resistance to treatment. And also in cardiovascular medicine, we can determine, physicians can determine the dose of warfarin genetically, and they can also predict the risk of side effects from statins. So personalized medicine is a reality. It exists. And it has been so advanced that some of these tools have been already tested in randomized clinical trials for their clinical benefit and their cost effectiveness. And as you may notice, all these publications are done at the New England Journal of Medicine because they are so important publications. But it's not a perfect world, and it's not a perfect field of medicine because there are several limitations and challenges. Complex diseases are probably the geneticist's nightmare because they are very difficult to investigate because they are complex. It's very difficult to replicate associations, and even if one does, these associations may not be applicable to all populations. But even if they are, the clinical utility of the test may not be very good, or the cost of the test may not justify their widespread use. However, these advances are very important, and are there any advances in common eye diseases? Yes, there are. This is a comprehensive review published just a few months ago, and it describes very nicely how this genomic research has provided a lot of insight into the pathophysiology of other complex diseases, such as AMD, diabetic retinopathy, and myopia. Genomics is the reason why we know now that inflammation is involved in the pathophysiology of AMD. We didn't know that before, and these pathways are important because they represent potential therapeutic targets. But what about glaucoma? So glaucoma is typically adult onset, it's complex, it's insidious, it's a neurodegeneration. From a pathobiological point of view, it's a progressive death of the retina and ganglion cells. But we do not know which one is the starting point, and there may be different starting points leading to the same result. Also, it's very variable, phenotypically speaking. It develops slowly along a continuum, so That's why it may not be always possible to tell whether a person has glaucoma or not. Maybe because the clinical signs are below the threshold, the standards that we use to define the disease. At the same time, some people with the same disease may have different phenotypes, but also people with different types of glaucoma may have exactly the same phenotype. And this is what makes things very complicated. But because they're complicated, and because glaucoma is a complicated disease, the best approach is probably to break it down into pieces, to study the traits of the disease. Intraocular pressure, corneal thickness, disc size, CD ratio are all traits of the disease, and they are easier to study and easier to quantify instead of saying these people have glaucoma and these people don't. So the progress so far involves familial linkage studies which have identified several loci and some genes. This is a new one. That's an interesting gene that seems to be implicated in the regulation of the intraocular pressure, and that's what makes it interesting. Then genetic association studies which have revealed loci in the DNA associated with the disease itself and with pseudoexfoliation, and then QTL analysis which has associated traits with areas of the genome. But there are many challenges. The first challenge is the heterogeneity of the disease itself, but there's not a lot we can do about that. It's just the nature of the disease. These studies require a lot of power, very, very large samples, and that's why all these consortia and international collaborations are necessary, but also meta-analysis can be useful because they combine different studies and they can increase sample size this way. And also, the imprecise definition of phenotypes is a problem, like using the dichotomous state of the disease, and for that, emphasis should be given on detailed phenotyping, especially when we're designing biobanks. And in this context, we have published a review paper a few years ago about biobanks, and about this has been neglected in the literature. But through this paper, we also presented an effort called GLOCOGEN, which is a pan-European genetic epidemiology network to provide a major resource for glaucoma in the future. The first steps of the study have been completed with the participation of 4 centers, and the Aristotle University of Thessaloniki, with Professor Fotis Topouzis as supervisor, is the coordinating center for the project. Also, in this year's ARVO meeting, we presented the results of a meta-analysis that we did on LOXL1, which is this major gene associated with pseudoexfoliation. We found big differences in different populations because Caucasians, Japanese, and Koreans are different. So for some populations, this is a risk factor. For other populations, the SNPs are protective. And also, we examined the association with this specific type of glaucoma, and we didn't find it. And this was recently also confirmed in the Thessaloniki Eye study, which is a large population-based study in Thessaloniki. We confirmed the association with pseudoexfoliation, but we were not able to confirm it with glaucoma, which means that this specific type of glaucoma, which is so common in Greece, is much more complex than we originally thought. So, what are the future steps? Future steps would be to identify more genes, more pathways, and to understand more about the disease. Optimally, to develop gene-based tests, which would be useful for the clinician, but they would also be useful in clinical practice and cost-effective. And then to develop strategies to treat disease-related events, because what we do in glaucoma now, we don't treat really the disease. We treat the intraocular pressure, which is the risk factor for the disease. And in conclusion, medicine seems to be changing. Using genomic information in clinical practice is a reality. Other doctors do it. It's just that we don't do it in ophthalmology yet, although there have been significant advances in common eye diseases, especially age-related macular degeneration. And the personalized approach in glaucoma is still an unmet need. However, based on all of that, it seems that it is a very, very realistic goal. Thank you very much for your attention.
I'm very proud for the level of Soul Lecture of Greece. And now we are going on. Thank you very much. Again, I'm saying I'm very proud. We are electing the best. And you are seeing the level of the young ophthalmologists of Greece is very high. We have to believe this. And now we are going on. Professor Kozoboulis, please coordinate the young ophthalmologist lecture, rapid fire lectures that will be in English. And again, I'm saying